Hello and welcome to lecture 6 of our course on stochastic modeling of biological processes. In lectures 4 and 5, we illustrated possible differences between the solutions of deterministic ODEs and the average behavior of the Gillespie stochastic simulation algorithm by discussing chemical systems with multiple favorable states and chemical systems with oscillations. In lecture 4, our examples included chemical system 2.1, and we observed stochastic switching between the two favorable states of the system. In lecture 5, our examples included the chemical system 2.9, and we showed that the stochastic model can oscillate even in parameter regimes where the deterministic model does not oscillate. What about if deterministic ODEs do not have such an interesting dynamics? What about deterministic ODEs with a single stable steady state? Do they provide a reasonable approximate description of the average behavior of the stochastic model? If our chemical system only includes the first order reactions or the zero order reactions, then we can prove that the classic deterministic ODEs provide the exact description of the average behavior of the Gillespie stochastic simulation algorithm. Considering higher order chemical reactions, a simple system with a single favorable state was studied in section 1.4. It is a system of one chemical species called A, which is subject to these two chemical reactions. We can analyze this system using the probability generating function to obtain that the stationary mean is around 10.13, which is not equal to the steady state of the deterministic ODE model, which is 10 for our parameter values. Obviously, the difference between 10 and 10.13 is relatively small, so we could still use the deterministic ODE model to get a reasonable approximate description of the average behavior of the stochastic model. In this lecture, we will discuss that even in the case of a single favorable state, we have to be careful when we use the ODE-based modeling. I will illustrate this using a simple example. We consider three chemical species A, B, and C in a container of volume nu, which are subject to the following six chemical reactions. Our presentation of this system will be based on the work presented in this paper. We will refer to A as a signal and to B as a product. In the first three reactions, the product B is produced from the intermediate species C and degraded. The intermediate C is produced with rate K1. The coupling between the production of the product B and the signal A is indirect. The signal A catalyzes the degradation of the intermediate C, which is described by this reaction. We will study how changes in the number of signal molecules A influence the changes in the number of product molecules B. We will show that the mean value of B depends not only on the mean value of A, but also on the fluctuations of the number of signal molecules A. To that end, we let the signal A evolve according to the production degradation system 1.9, which we studied in section 1.2 in detail. The corresponding deterministic description can be written as a system of three ordinary differential equations for concentrations. The first differential equation describes the production degradation system for the signal A. The right-hand side only depends on the concentration At because the chemical species A is not modified in any of these reactions. On the other hand, the concentration of the product B does depend on the concentration of A because A is included in this term K4AC. Our goal is to compare the deterministic and stochastic description of this chemical system. To do this, we have to transform concentrations into numbers of molecules. Starting with the ODE system 222 till 224, we multiply these ODEs by the system volume nu to obtain the following system of ODEs for approximate numbers of molecules. They include six parameters: K5 nu, K6, K2, K3, K1, and K4 over nu. These are exactly the same six parameters which we need to specify to apply the Gillespie algorithm. The values of parameters K1 nu, K2, K3, and K4 over nu are given in the lecture notes. We assume that the value of the rate constant K5 is abruptly halved at time 10 minutes. That is, we choose the rate constants of the production degradation subsystem 221 by this formula. In figure 2.6 on page 43, I plot five realizations of the Gillespie algorithm together with the solution of the derived system of ODEs. The left panel shows the time evolution of the signal A, and the right panel shows the time evolution of the product B. We observe that the average number of signal molecules A halves at the time 10 minutes because of the change of the production rate K5. However, the average response of the number of product molecules B to the change in the signal is significantly larger than the response predicted by ODEs. The steady state of the ODEs jumps from 100 to around 200, while the average of stochastic simulations jumps from 100 to around 300. There are different ways to interpret this result. First, we will show that the mean value of B depends not only on the mean value of A, but also on the fluctuations of the number of signal molecules A. To illustrate this, I will remove fluctuations of A from our simulations. To do that, I will not consider these two reactions. I will simulate a reduced chemical system without a production degradation subsystem for A. The results are presented in figure 2.7 on page 45. They have been calculated by the Gillespie algorithm applied to the following system of four chemical reactions, where we directly impose the time evolution of the signal A in the following form. In particular, the left plot has no signal fluctuations. We observe that the responses of both the deterministic and stochastic models are similar. The deterministic ODEs seem to give the average behavior of the stochastic model. And we can prove this. Our reduced chemical system contains four chemical reactions. The fourth reaction is written as a second-order reaction. The signal A catalyzes the degradation of the intermediate C. Such a reaction can also be formally written as the degradation of C with the rate K4A. In both formulations, one molecule of C is removed from the system whenever the reaction occurs, and both formulations have the same propensity function. So this is just an equivalent formulation provided that we are happy to put a time-dependent rate above the arrow when writing this chemical reaction. In the case of the reduced chemical system, we have no fluctuations in A, and At can be assumed to be a constant. Then, reaction 237 is the first order chemical reaction, degradation, which was studied in section 1.1 as the chemical reaction 1.1. Therefore, our reduced chemical system only contains first order and zeroth order reactions. Using techniques from chapter 1, we can show that the deterministic model provides an exact description of the average behavior of our reduced stochastic chemical system. In particular, the result presented in figure 2.7 is confirming this theory. The average behavior of presented stochastic simulations is exactly described by ODEs. So this reduced model is one of these simple chemical systems when we do not have to do any stochastic simulations because we can fully analyze them. However, it is actually very informative to do such a simulation. If you try to write your own computer code for stochastic focusing, then one possible way to test your code is to comment out the production degradation system. Then your computer code will describe the reduced chemical system, and you can test whether it correctly works by comparing it with this theory. In the case of the reduced chemical system, the average of stochastic simulations jumps from 100 to around 200. If we add fluctuations to the signal, then the average of stochastic simulations jumps from 100 to around 300. In particular, if we assume that a certain process can be switched on inside a cell if the product B crosses a certain threshold, say 250, we observe that this is possible for the fluctuating signal, but not for the signal without fluctuations. This would be one of the ways to interpret our results. However, our main aim of Chapter 2 is to compare deterministic and stochastic modeling. So let us try to understand why DODs do not seem to correctly predict the average behavior of the stochastic model. I have highlighted here the second order reaction A plus C goes to A in red. If this reaction was not part of our system, then the system would only contain first order reactions or zeroth order reactions, and we would not expect any differences between deterministic and stochastic modeling. However, the presence of fluctuations of the second order chemical reaction is not a sufficient condition to obtain such significant differences between the stochastic and deterministic descriptions, as presented in figure 2.6. Indeed, we have already mentioned the dimerization example at the start of this lecture, for which the corresponding deterministic equations provide a reasonable approximate description of the average behavior of the system. To illustrate this, for our stochastic system, I will change some parameters. Figure 2.6 was calculated using these four parameter values given in equation 228, and we specified the rate constants K5 and K6 as in equation 229. Next, we keep the same values of K5 and K6 as in equation 229, but we modify the first four parameters as in equation 238. The results are presented in figure 2.8 on page 47. Since the rate constants K5 and K6 are the same as before, the left panel is the same. However, we also observe that there is no stochastic focusing. There is no jump of these stochastic trajectories to 300. The deterministic ODEs look to be doing a very good job when describing the average behavior of the stochastic model for this new set of parameter values. So, what is different between the parameter sets 228 and 238? I chose them in a way that the deterministic dynamics of the signal A and the product B stays the same. However, I have not yet mentioned anything about the intermediate chemical species C. Indeed, these two different sets of parameters give different dynamics of C. The results are presented in figure 2.9 on page 48. The left panel is for the original parameters and the right panel is for my new parameters. You can see that C is present in low copy numbers. Only 0 or 1 molecules of C have been in the system at any one time in the original simulation, when significant differences between the stochastic and deterministic description of the product B were reported. On the other hand, there is more molecules of C in the system for my new set of parameters. In the left plot, we have a very low number of the intermediate molecules C in the system, which makes the ODE model inapplicable. Therefore, second-order chemical reactions, together with low copy numbers of some chemical species, can lead to significant differences between the average behavior given by the stochastic model and by the corresponding approximate deterministic ODEs. We note that in our illustrative system, the important quantity, the signal, which we observed, was the product B, which had molecular abundances of the order of hundreds, which could be considered large enough for some differential equation modeling. However, we still reported significant differences between the stochastic and deterministic description of B, because the intermediate chemical C was presented in low copy numbers. This conclusion is good to have in mind when modeling more complicated biological systems. This is the system of two mutually repressing genes, which we discuss in detail in lecture 4. When I mentioned that our modeling of the protein production was highly simplified. This highlighted reaction describes both transcription of DNA into messenger RNA and translation of mRNA into protein. In particular, we could write a more complicated model, introducing the numbers of mRNA as additional variables and replacing both highlighted reactions by these two reactions with new rate constants. I denoted them as K13 and K14 to distinguish them from the parameters which are already in the model. DNA is first transcribed into mRNA, which is then translated into the corresponding protein. Interestingly, there is some analogy between this model of mutually replacing genes and our stochastic focusing example. There could be a very few copies of specific mRNA molecules inside a cell. So mRNA could not only be an intermediate species, but it could also be in low copy numbers, like the intermediate chemical C in our previous example. In particular, even if we are not interested in intermediate mRNA, but in the final protein products, our results could differ depending whether mRNA is explicitly included in our modeling or not. This brings me to the end of lecture 6. We have discussed section 2.3 on stochastic focusing and showed that the differences between deterministic ODEs and the average behavior obtained by the Gillespie algorithm can be significant for systems with low molecular numbers of at least one chemical species. Please read section 2.3 on pages 42 to 49 of the lecture notes before watching the video of lecture 7. Thank you for listening to lecture 6 of our course on stochastic modeling of biological processes. Bye bye.